Hello everyone, welcome to Science Made Easy. In today's video, we shall study about cell cycle and cell division. The cell cycle is a bit complicated process, so I will start by explaining different terms related to cell cycle and cell division. Terminology related to cell cycle and cell division. Firstly, let's see what is a haploid. The haploid refers to half of the somatic chromosome number. It is denoted by N. This haploid chromosome number is usually seen in gametes, so it is called as gametic chromosome number. The next term is diploid. Diploid refers to the somatic chromosome number. This is denoted by 2N. The diploid chromosome number is usually seen in zygotic or somatic cells, so it is called as zygotic or somatic chromosome number. In this picture, you can see diploid and haploid cells. In diploid, there are two copies of each chromosome, and in haploid, there is only one copy of each chromosome. The next term is chromosome. When observed under light microscope, chromosomes are darkly stained rod-shaped structures. This shape is clearly seen only during metaphase stage of mitosis. The next term is centromere. Centromere is also called as primary constriction or kinetochore. The centromere is the region of the chromosome with which the spindle fibers are attached during the metaphase stage. This is the structure of the chromosome, and you can see the centromere labeled in this picture. Now let's see what is a chromatid. It is one of the two distinct longitudinal subunits of a chromosome. Here you should know about sister chromatids and non-sister chromatids. Sister chromatids. The chromatids belonging to one and the same chromosome are called as sister chromatids. The chromatids belonging to homologous chromosome are called as non-sister chromatids. I will explain by showing a picture. These two chromosomes are called as homologous chromosomes. Here, the chromatids from the blue one are called as sister chromatids, and the chromatids from red ones are called as sister chromatids. Now, if you consider one chromatid from blue one and one chromatid from red one, these two are non-sister chromatids. This is all about the terminology. Now let's move on to the topic, cell cycle. Now what is the cell cycle? It is the period in which one cycle of the cell division is completed. The cell cycle has two phases. These are interphase and mitotic phase. In the further video, we shall see the details about these phases. Firstly, interphase. Interphase is usually called as DNA synthesis phase. It usually occurs after the telophase stage of previous mitotic division and before the onset of prophase stage of next one. So this interphase has three subphases. They are G1 phase, S phase and G2 phase. Let's see the details of these phases. G1 phase is also called as gap1 phase. This is the pre-DNA replication phase, and in this phase protein and RNA synthesis takes place. The next phase is S phase. It is also called as synthesis phase. Here the chromosome and DNA replication takes place. The next phase is G2 phase or gap2 phase. In G2 phase, considerable RNA and protein synthesis occurs. It is the post-DNA replication phase, and some proteins synthesized in G2 phase are essential for the entry of cell into mitosis. As you can see, this is the picture of the cell cycle, and as seen in the picture, G1 is the longest phase and M is the shortest phase of the cell cycle. This is all about cell cycle. Now let us see cell division. Cell division is the process of formation of new cells from pre-existing cells. Now you may wonder why the cells have to be divided. To know that, we should know the functions of the cell division. The cell division produces daughter cells. These daughter cells helps in functions like growth and development, regeneration of damaged tissues, replacement of old organs and tissues. The next function is it keeps the cell size limited and helps in tissues and organs differentiation. To understand the tissues and organs differentiation, now let us consider if the growth was only due to increase in cell size, then there would be no different tissues and organs today, and there would be no functions those tissues and organs performing. So cell division and differentiation is important for existence. The events during cell division. These are karyokinesis and cytokinesis. The division of nucleus is called as karyokinesis and the division of cytoplasma is called as cytokinesis. The cell division is of two types. They are mitosis and meiosis. In this video, we are going to learn about mitosis, the different stages of mitosis, occurrence and importance of mitosis. Mitosis. Mitosis was discovered in 1879 by Fleming and the term mitosis was given by him in 1882. Now let us see about mitosis briefly. In mitosis, the parent cell undergoes some changes and produces two daughter cells. Here you should remember that in mitosis, the number of daughter cells produced are 2 and these daughter cells are identical to the parent cell. Occurrence of mitosis. Mitosis occurs in somatic tissues. It occurs in spores. The zygote, which is formed from fusion of two gametes, undergoes cell division, that is mitosis, to produce embryo. Mitosis helps in wound healing and damage repair. Stages of mitosis. Mitosis occurs in different stages. These stages are prophase, metaphase, anaphase and telophase. This image shows the different events occurring during different stages of mitosis. By the end of the video, you will clearly know what are the different events occurring during different stages of mitosis. Without any delay, let us see the stages of mitosis. The first stage is prophase. The prophase starts immediately after G2 stage of interphase. In prophase, there is chromosome condensation. The chromosomes are present as chromatin material throughout the interphase and they don't have any distinct shape. As the mitotic phase starts, the chromosomes starts condensing and attain a distinct shape. In early prophase, these are thin and uncoiled. In the mid-prophase, these are shortened, coiled and more distinct. In the late prophase, these are more short, thick and double when compared to early and mid-prophase. At the end of the prophase, the nuclear membrane starts disintegrating and the nucleolus disappear. Here make a note that the end of the prophase is sometimes called as pro-metaphase because it marks the end of the prophase and beginning of metaphase. Here in early prophase, the chromosomes are uncoiled and not distinct, and in the late prophase, the chromosomes are distinct, coiled, and the nuclear membrane start disintegrating. The second stage of mitosis is metaphase. In metaphase, the chromosomes are in shortest and thickest stage. As the chromosomes are more distinct during this stage, most of the studies related to chromosomes are done during this stage. Now, what are the different events occurring during metaphase? Firstly, the chromosomes align at the equatorial plate. The spindle apparatus is organized and the spindle fibers are attached to the centromere of the chromosomes. There is no nucleolus during this stage and the nuclear membrane which has started to disintegrate during the end of the prophase disappears completely during this stage. In this picture, you can see the different stages of metaphase. The chromosomes are aligned at the equatorial plate with attached spindle fibers from the spindle apparatus. The third stage of mitosis is anaphase. During this stage, the centromere separates longitudinally and the chromatids move to the opposite poles. If you see the image, you can see the sister chromatids are separated and are moving to the opposite poles. The next stage of mitosis is telophase. The events occurring during telophase are the spindle tube disintegrates, nucleolus forms, nuclear membrane forms, and the chromosomes uncoil and unfold. In the image, you can see nucleolus and nuclear membrane around the divided chromosomes. By the end of the telophase, the nucleus is divided and now the cytoplasm has to be divided. The division of cytoplasm is called as cytokinesis. Cytokinesis is different in plant cell and animal cell. In plant cell, a cell plate is formed in the center of the cell that grows towards the periphery and divides the cell into two daughter cells. And in the animal cell, a furrow grows from the periphery of the cell towards the center and divides the cell into two daughter cells. In these images, you can see the chromosomes in the nucleus have completely uncoiled and are no more distinct. With the completion of cytokinesis, there are two daughter cells that are similar to the parent cell. These are the products of mitosis. Now let us see the importance of mitosis. Mitosis helps in development of zygote. It helps in development and growth of different tissues and organs. It helps in maintaining purity because the daughter cells are similar to the parent cells. In animals, mitosis helps in replacement of old tissues with new ones. In plants, the spores undergo mitosis and enter the gametophytic stage. In vegetatively propagated crops like sugarcane, potato, etc., the mitosis helps in asexual reproduction. This is all about mitosis, its importance, and its occurrence. Remember that all the stages of cell division and classification we have seen today are just for our convenience in discussion and description. In reality, cell division is a continuous process which gradually moves from one stage to the other, and there are no clear-cut boundaries between these stages. This is for today. In next video we shall see what is meiosis, stages of meiosis, importance of meiosis. For now, please make sure to subscribe to Science Made Easy, and your feedback will be always useful. Thank you for watching the video, and please keep supporting for more such videos.